for Innovation in Global Health Technologies, which is called the acronym CIGHT or CIGHT, joined the Institute for Global Health as one of our 10 centers. CIGHT is a joint center between the Department of Biomedical Engineering at the McCormick School of Engineering and Applied Sciences and the Havey Institute for Global Health at Feinberg School of Medicine. The center's mission is not merely to develop, but also to bring to market comprehensive, context-appropriate healthcare solutions that integrate seamlessly with the lives of patients and medical practitioners in the developing world. Today, I'm pleased to introduce our two presenters for today's webinar, co-directors of the Center for Innovation in Global Health Technologies, Dr. Matt Glucksberg and Dr. Sally McFall. Dr. Glucksberg is a professor of biomedical engineering at Northwestern and co-director of CIGHT. His technical expertise is in tissue mechanics, microcirculation, and optical instrumentation. He is co-founder of Northwestern's Global Healthcare Technologies program in Cape Town, South Africa. And he just returned from there yesterday. He's co-director of a Master's in Science certificate program in Global and Ecological Health. And he's working with Feinberg to develop biomedical engineering degree programs at 3 universities in Nigeria. Dr. Glucksberg is a member of the College of Fellows of the American Institute of Medical and Biomedical Engineering. He's a fellow of the Biomedical Engineering Society and serves as a founding board member for the Northwestern Global Health Foundation. Dr. Sally McFall is a research professor of biomedical engineering at Northwestern and co-director, along with Dr. Glucksberg, of CIGHT, as well as the co-director of the administrative core, MPI of CETHAM, which is Technologies in Health, and director of the Technology Development and Refinement Core. Dr. McFall has extensive experience in leading point-of-care technology development projects for infectious diseases, such as HIV, TB, hepatitis, COVID, and others designed specifically for low and middle income countries. She recently founded a startup company with fellow Northwestern faculty, Dr. David Kelso and Kara Palamountain, called Minute Molecular Diagnostics, to market a point-of-care testing device in settings with a goal of developing applications to expand that technology to low resource centers. She received her PhD in microbiology and immunology from the University of Illinois. So welcome, Matt and Sally. We all look forward to hearing your talk today.
Thank you, Bob, and thank you for giving us this opportunity to tell everyone about our Center for Innovation in Global Health Technologies. So CIGHT has a number of different focuses. We have a research and development arm where we're focused on diagnostics and neonatal and maternal health. And we also have an education arm where we're working on engineering for health, educating our Northwestern students, and also building capacity in low and middle income countries for both biomedical engineering and also product development. The site is located on the Evanston campus of Northwestern, and we're located about right up in this where all the North Campus science buildings are located. We are in Hogan Hall, which was designed by the architect Walter Netsch, and it's a hexagon. And fun fact is that when this building was built, it was the most eastern building on campus, and you can see that they decided to just have these narrow little windows on the very corners because apparently Walter Netsch thought that scientists were inward thinking. But anyway, that's my fun fact for the day. CIGHT was founded by Dave Kelso and Matt Glucksberg. Dave Kelso was also the founder of the CIGHT Product Development Laboratory. And this was founded on money from the Bill and Melinda Gates Foundation, one of the first of their Grand Challenge Awards. And it followed very soon after George W. Bush instituted the PEPFAR program, which has funded giving medications for HIV to 25 million people. Lives have been saved in low and middle income countries. And so if you have medication for HIV, you also need to have treatment monitoring to be sure that the virus isn't developing mutations and then becoming resistant to the medication. And so our first projects were on developing HIV viral load tests. So now I'm going to have Matt tell you a bit more kind of about what the education arm of the site is involved in.
Yeah, thank you, Sally. We are a center and not just a laboratory because we try to take a comprehensive view of providing technologies for global health. Technology alone won't really solve many problems. It needs to be integrated into health systems. You need to understand the entire approach. Part of what we do is also we educate students in global health, but we also use them as agents for change in global health. There are many times our eyes and ears, creative people who are developing products, or more often contributing to the development of products. We're based, most of our operations are based in the Cape Town area. This is a view of Cape Town. It's a spectacular setting for a city. It's a playground for the rich in many places. If you look you see Table Mountain there, and to the left of Table Mountain, you see Devil's Peak. And just down the slopes of Devil's Peak on the other side of the mountain is, next slide, the University of Cape Town, where we're based. Again, spectacular setting. But if you turn around 180 degrees and go down the hill, drive down the hill for about 10 or 15 minutes, next slide, you're in the townships. The townships of South Africa are among the most populous. The biggest one outside of Cape Town, it's sort of near the airport, is Khayelitsha. There's probably a dozen other townships, and more than 1 million people live there. And you can see the conditions there are nothing like the conditions in the city. This is a sort of sandy, poor soil. It's not the worst of conditions. There's shacks. This is the kind of place where people live in all over the world. You can see this particular township is turning into a. This is an old picture. If you go there now, you'll see some cinder block structures turning into a more of a middle class, working class area. You can see telephone poles there. There is electricity. There's some sanitation, but it's really a substandard living. If we keep, this is a market in Khayelitsha. You can see it's a lively place. A lot of these places are probably worse than you might expect and better than you might expect in different aspects. It's in many ways a thriving community, but the healthcare needs are great. Next slide. We've also worked at Stellenbosch University. Again, a stunning setting. Next slide. It's a wine growing region. The workers live nearby, and their conditions, next slide, are a little bit more rural, but still very substandard. Again, you see there are power lines here. People do have cars, but it's a poor area with great health needs, and they're somewhat different than the ones in the larger townships near Cape Town. This is about an hour's drive from the University of Cape Town. So there are two universities that are very close together in the Cape Town area. Next slide. We also do some work in Lagos. I'll talk about this maybe a little bit later. Again, Lagos is many ways a modern city. It's the largest city in Africa, 21 million people, so I'm told. Next slide. This is also Lagos. The world has changed in many ways. We're not talking as much about developing world and developed world. The world, around the world, many countries have developed and have not. You see development and underdevelopment in the same place. The world isn't separated into the developed world and the developing world. Really is much more. The lines have been blurred. Next slide. I can always show Chicago as well. Beautiful city, lots of culture, great architecture, art. Next slide. We have our problems as well, and it's a matter of degree rather than it is a matter of of difference. What that means is that the technologies, the underlying technologies that are available to us are available in South Africa, are available in Nigeria, and indeed are available around the world. Cell phones are everywhere, cars are everywhere. We have the opportunity, a little bit different than it was 30 years ago, to have the infrastructure to develop technologies, but they still need to be appropriate technologies for the healthcare needs of the place. Next slide. So this is, I want to make the point that the world has changed, but that the disease burden in LMICs, low and middle income countries, is profoundly different than ours. The division between the developed and developing economies have really become blurred. And I also want to make the point that things are getting better in most places. And I want to make that's something that many Americans, Europeans, they feel that places like Africa keep going, we keep pouring money in and nothing changes. The fact is that a lot changes. PEPFAR was enormously successful. If you look at under 5 mortality, this is before COVID, you can see that it falls dramatically. And this was not an accident. This was a plan. This is the way that this is the Millennium Development Goals. This is a concerted effort by the world to make things better for children. Next slide. Children are getting vaccinated. The vaccination rates, again, this is just until 2018, but they're climbing, and they're climbing fast. Next slide. People, this is another concern. We're going to be testing. We want to test people for HIV. They're getting tested, or we think we have some devices that'll improve that testing. Question is often, do people, if you test, can you treat? And the answer is yes. These numbers are going up. People who have HIV are on antiretroviral drugs, and they're staying on them, and they're surviving. It's becoming, the disease is being managed. Next slide. So I want to talk a little bit about how we use our students as we educate our students in developing some of the technologies. Some of them are very sophisticated. Some of them are very simple. We're not looking to develop technology. We're looking to develop solutions to problems. This is a group from, I think this was the crowd just before COVID at the Cape of Good Hope. These are all Northwestern students, somewhere in the middle of their program on a cold and blustery day down on the Cape. Next slide. So going way back, we started thinking about global health back in about 2005. David Kelso was teaching the senior design course. Biomedical engineering was becoming very popular. We just had too many students for him to handle on his own. So I was asked to join him, and I jumped at the chance. At the same time, and a couple of years later, Dave Kelso decided to put in a grant to the Bill and Melinda Gates Foundation in the first Grand Challenges. This is about 2005, I think, 2004. And he had the idea that there's a lot of great technologies, that a lot of great projects we could use some engineering technology for, and we should give them to our class. The first project we did, and this I show maybe as a little penance for our arrogance at the time. We've learned a lot since then. So let me just describe this. This was an infant incubator, and at the time, everyone told us, we were working with a medical anthropologist. Everyone told us that they really need infant incubators, but we had not been there to visit yet. We didn't know what they needed. So I gave this to a group of students, and it was really one of the best groups I've ever had. They did calculations, they did engineering calculations, simulations, they built prototypes, they did experiments, they measured. It was really an excellent group. They decided to go with phase change materials because we didn't want to have to plug this thing in, because we heard there were power problems. We had antimicrobial coated plastic. But if you take a look at the lower left corner, if that looks like a laundry basket from Target, that's because it is a laundry basket from Target. And if you wouldn't put your baby in a laundry basket from Target, we don't want to ask poor people to do that. This, in retrospect, was a shortcoming of our vision. We didn't understand what was going on in poor countries. This really is not an acceptable solution. We decided that we really did need to get to South Africa. So we started this program. It's a full academic term in South Africa. We have typically about 15 or 16 students go. They're not all biomedical engineers. We brought this device to South Africa. Next slide. And they were very kind to us, not at all patronizing, but they did tell us to put that in the corner with all the other homemade incubators. And they took us into the clinics and showed us what they really do. And what they're doing there was really innovative. This is now spread to some other countries. Kangaroo mother care is practiced around the world, but not in the same way it's practiced in South Africa and a few other places. These are premature infants. These are preterm infants. They might be 1,600 grams. They're placed on the mother's chest and they're wrapped up and kept warm. The mother's chest keeps the babies warm and moist. If the baby starts overheating, the mother's breasts will cool the baby. It doesn't work on a mother's back. It doesn't work on a man's chest. This was a technology we were not aware of. In the lower left panel, you can see one of these babies. They are too small to feed themselves. That's a feeding tube coming out of the mouth, and the mothers maintain the feeding tubes. They do all the feeding. They do all the work. It's very stressful on the mothers, but this can go on for 6 weeks or 2 months in the hospital as the babies gain weight. And it turns out they gain weight faster. There are other indications of babies do better, but there still are problems here. And this is what they posed to us. They said, forget the incubator. We don't need that. What we need is some monitoring of the babies. These babies need CPAP. These babies need phototherapy, and these babies need to be monitored for apnea of prematurity. That is, the babies can stop breathing spontaneously. And if the mom's awake, they can kind of shake them and get them breathing again. That works well enough. But this is 24 hours a day, 23 and a half hours a day. The babies are on the mom's chest. We've been there when this has happened. The babies can just stop breathing and the babies can die, especially if the mother's asleep. So we needed a monitor for the babies. Next slide. So what we really needed was a neonatal intensive care unit, a NICU built on the mother's chest. And that was the first project we started with. Next slide. Now, these projects don't get done in one term. The students go, so many students worked on this project, but I wanted to highlight a couple of students. This is James Fox and Paige Cowles. Paige was actually, Paige was not a formal member of the program, but James came and worked on this project with a company called Infant Trust. He was a mechanical engineer. He is a mechanical engineer. And he went and stayed for a full year there to work on these projects. Paige was his fiance at the time. She's a nurse, but they worked together on this project and others. Next slide. And developed a unit that can measure that can monitor the infant's breathing. That's on the left, and a phototherapy blanket that can be wrapped around the baby to treat the jaundice that comes with preterm birth. Next slide. And just wanted to show where they are. Well, not really now. This is James. James has been very active. They co-founded something called OBS Medical Design to work on similar types of projects. He's currently vice president of business development of Starfish Medical in Boston. And that's a picture of Paige, a remarkable person. She volunteered. This is her in 2014, 2015 in Sierra Leone, where she volunteered for 10 weeks as a nurse during the Ebola crisis. We've had over 200 students come through, and I could tell stories about many of them, but I just wanted to highlight a couple. This is the next slide, please. This is another project that's close to our heart. This is really in diagnostics, and this is a pie chart of how TB sample collection can fail. About 10% of TB sputum samples. Sputum is the phlegm and mucus that comes up when you're really sick. And if you're sick with TB, you can usually produce this, and you put it into a specimen cup. You don't do it under observation. Nobody wants to be in the room with you when you're coughing up sputum for TB diagnosis. So they put you in a room by yourself, and you're instructed to produce between 1 and 2 ml of sputum. And if you look at this chart, that big green wedge is insufficient specimen. It means they didn't give you enough. And why is that? So that counts for then about 8% or so of the samples, and that's an expense. Those people have to come back. That has to be redone. Can we fix this? And what is the problem? Next slide. These two students, that's Darren Jacoby and Jillian Geary. Darren was a BME student. Jillian was industrial engineering. Next slide. And what they and their client recognized was that this is a sample cup. You're all familiar with it. It's usually used for urine, but this is what they had, and this is what they would give the patients to go and deposit their sputum. Most people don't have trouble producing sputum. Children do. They have difficulty producing it and getting into the cup. And HIV patients do, which is really a very large chunk of the TB population in South Africa, partially because of the muted immune response due to HIV. So they would not produce enough. But you can imagine 1 ml in this 100 ml cup is very hard to know how much you produced. So the idea was to produce a different cup. Next slide, please. And this was their idea. Over many iterations, they worked with a company in Stellenbosch called Sinapi. They do injection molding. And if you look carefully at the bottom of the cup, there's 2 blue lines, a minimum and a maximum. Now the patients know how much to produce, and they eliminated those rejected samples. Some other improvements because of processing. We want to make sure these infection, these cups don't leak like the previous cups I showed you. And they processed a lot of these, so there was repetitive stress injury happening to the technicians because they were unscrewing and rescrewing on these caps. So this is a quarter turn cap that goes on. So it's a very well designed specimen cup. It's more expensive. Instead of 4 cents a piece, it's more like 15 cents a piece. But the money saved in not having to repeat tests more than makes up for it. Now this is on the market and has been adopted by a number of facilities. Next slide, please. And just to let you know where some of our students go, they're really, they come to us really interested in health, and they leave, I think, even more interested from the experience in South Africa. There's Darren there. He now runs a company, the CEO of Protosthetics in Fargo, North Dakota, and they 3D print sockets for prosthetics for amputees. Next slide. And Jillian, who was a industrial engineer, went to work, I think not coincidentally, for a diagnostics company. She worked there until recently, and she's now at Berkeley getting her MBA. Next slide, please. Another area we look at is capacity building. We're over in Africa a lot, and we try to bring them here. We have colleagues there. This is how we stay in touch with what's really going on. And we've been promoting biomedical engineering as a discipline on the continent. We've consulted and had done all sorts of things with all sorts of universities, but there are 3 main ones at the University of Ibadan in Nigeria, University of Lagos also in Nigeria, and Technical University in Bamako, Mali, where faculty, myself and others, are coaching. I think is maybe the best way to do it. They're doing it, but we're helping out to establish biomedical engineering as a discipline. What I have, the picture here is the biomedical engineering building in Ibadan, Nigeria. They built a building. They have a full department now. That was started in about 2015 or so, and is going very strong. Collaborations with other universities, of course, and we're very excited to host visiting scholars from all these places, and hope we get some more. They really contribute. They do mentored teaching here. They come up with ideas, and it keeps the collaborations going. Next slide, please. I'm going to transition over to back to Sally, where she'll talk about the research in the department. One person I haven't mentioned yet is Kara Palamountain. Our collaborations are certainly with the medical school, but they're also with the business school. Everything we do needs to fit into the system, the medical and healthcare systems in the countries we work in. And the person who really does this best is Cara Palamond and her Kellogg students. This was back in 2007 when we were just getting going. Quite a bit has changed, but the process hasn't. And I'll tell you what some of the results of this market research is after. Well, Sally will tell you what the results of this market research is and where it's led the research and the product development at site.
Thanks, Matt. So yeah, it's this is a picture of Cara playing kind of a game with some people that are healthcare providers about what kind of test would they like to have in their clinic the most. And so they have, they rank them. But what she did, she probed their answers, and what she found was that they didn't ask for example, rapid viral load testing and rapid TB testing, because they just didn't even think they were possible. So they put the rapid CD4 test because they thought that one was the least possible. So it's just kind of interesting how you have to probe deeply and find out, what people are thinking. And that kind of gives you a really good idea of what the needs are. So what came out of that was that we found that we really thought molecular testing for diseases such as HIV and tuberculosis was kind of at the top of the list. And we spent time developing different products and coming to a certain place. And then we kept having lessons learned. And then we were able to put the pieces together in 2017 to start to develop DASH, which is a fast PCR platform that can do a number of different sample types. And so Dave Kelso, Cara Palamond, and I are the co-founders. And as we grew and started to need more business functions, we added Wally Narajowski and Mark Minkus in operations. And so we were developing DASH and before COVID hit, and that was more for HIV viral load and also for kind of doctor's office types of tests. And then when COVID hit, we realized that we should pivot for this and try to develop something that could be placed in a lot of different settings to give a rapid, high quality result that is equivalent to what you would get if your test was run in a big central lab. Our goal was always have the test be 15 minutes or less so that it could be, the result could be provided within a clinical encounter. We use the gold standard quantitative PCR as our detection method. And when I say multiplexing capability, what I mean is that more than one test can be run on the same sample at the same time. So for example, we're developing a test that can detect COVID, flu, and RSV. So if you have similar symptoms, you can distinguish which disease you might have. The test cartridge contains all the reagents that are required, and it's designed to be really easy to use. And so when I say designed, what I mean by that is a cross-functional team of biologists, chemists, many types of engineers, software engineers, all participated in the development of the co-development of the DASH cartridge and the DASH analyzer. So we had biologists that worked on what kind of chemistry or reagents that go inside the cartridge. We had manufacturing engineers that worked on how to make this cartridge. How can we automate the manufacture of the cartridge so that we can make thousands a day? And with the analyzer, we had, engineers that worked on the software, on the electronics, on the firmware, which is, as far as I can tell, like how the instrument kind of regulates and defines, tells its different parts what to do. The enclosure, DASH connects to the cloud, so we have cloud software. So there's many different people have worked on DASH in collaboration with the scientists and engineers here at site. And then we also have an integration team of people that understand how the cartridge works and how the analyzer works, and make sure that the analyzer will perform all the steps of the test so that we can get an automated result. So last year, the DASH COVID test received emergency use authorization from the FDA for testing at the point of care. So that means by non-laboratorians, so healthcare workers can perform the test. We don't need it to go to a central lab. And I've already mentioned that the test is extremely easy to use, but I just think this is a great illustration here. Basically, the specimen is collected. It's just a nasal swab, goes into patient, puts a nasal swab into their nostril and swirls it around. And then it gets placed right into the cartridge, and it's broken off at this. There's a part of the swab that's made to be broken off easily, kind of like the way we can open chips. There's like a tear, there. And so you break off the swab, close the cap, and put it into the analyzer, and 15 minutes later, the result will appear on this touch screen. It also connects to the electronic medical records, and you can have a printer, so a printed result will come out. And so I thought you might enjoy just seeing what happens on the inside of DASH. So the DASH cartridge, as I mentioned before, comes fully loaded with everything that's required to run the assay. And that includes this particular thing called the liquid transfer capsule. And I'm sorry, but I think my video might not be in the beginning, but I'll fix that. So it comes with all the chemicals that are required for the test, and they are stored dried in the chambers where they're used. So here we can see that the pump is picking up the liquid transfer capsule, and it moves liquids around, and here it's rehydrating the magnetic particles. So we bind the RNA, the viral RNA, to those magnetic particles. And that helps us concentrate the RNA from the sample. We then just take those magnetic particles with this magnet right into our PCR chamber. And now we're doing our PCR reaction. We do 40 cycles of, it's a heating at 68 and a heating at 95, and those lights that flash right there, that's the fluorimeter reading the signal. And so after the 40 cycles, when the test is complete, the result will come up on the screen. As I mentioned earlier, the DASH cartridge wasn't designed specifically for COVID. It was designed for a number of different sample types, swabs, both respiratory, but also urogenital, also whole blood and plasma, and urine and saliva. And as I mentioned before, we have new tests in development. We have tests that can be used throughout the world, for example, for flu A, flu B, and COVID, strep A, sexually transmitted diseases, and some that will be more in use in low and middle income countries, especially tuberculosis. Of course, HIV and hepatitis C are global problems, but most global problems have a greater impact in low and middle income countries. And we also developed a test for monkeypox. So I wanted to acknowledge all the people that have participated in this project at site, but I also wanted to point out that other parts of the Institute for Global Health participated in this project. So the Northwestern RADx clinical team is made up of many of our physicians and other employees at the Institute for Global Health who are infectious disease clinicians, and then we also, I wanted to go to this slide to acknowledge our funding. And something that I really wanted to point out is that when we first had the idea to develop DASH, we received funding from the Institute for Global Health Catalyzer Award, and we also received an award from McCormick. And these small awards allowed us to create a proof of concept that we were able to then attract angel investors to get us going so that we could start to make prototypes, so that we could then be eligible to receive funding from the NIH through the RADx program to develop tests for COVID and to scale up our manufacturing. And so these kind of seed money really can provide the basis, the foundation for growing and successful projects. We're ready for any questions.
Okay, thank you, Matt and Sally. I'm just going to run through some of the questions that our viewers sent in and have you guys go through them. We've got about 20 minutes to go through this, and if there's any questions we don't cover, I'm encouraging the viewers to contact us by whatever method Jen has set up for you, and we will try to answer your questions later. Matt, I'm going to give you the first question that one of our viewers sent in. Who are the best people to ask in the countries within which we work about what types of innovations or solutions are actually needed?
Yeah, well, we work with quite a few people. We've been there for 17 years now. We return every year with students, and we make additional trips. So we're in constant contact with our collaborators, and many of the collaborators have been with us since the beginning. So it's their needs. It's clinicians. They're the obvious ones. They have a problem, and we keep in contact when they come up with ideas. Some of the less obvious people might be administrators, IT professionals. We've worked with, particularly in the neonatal area, where they need to improve techniques. One particular area that we worked on for several years was improving what's called flash pasteurization, a method to deactivate HIV in breast milk. Our collaborators had developed a method to do it. It wasn't working very well, so we helped standardize it. We helped measure temperatures. These were fairly routine measurements, fairly easy solutions, but someone had to examine it from a technical point of view. Our students were very able to do that. So it's really keeping in communication with the people who work there and understanding not just the technology, but how the technology is used and how it fits into the healthcare system in South Africa.
Great, thanks. And Matt, you certainly have a lot of partners in these sites. How do you actually get yourself and the team together to assess the unique needs of each individual community?
Well, so we have our people. We have the communities that we're involved in. We can't spread ourselves too thin, but we're really fortunate to have Cara Palamountain and her Kellogg students who can do a very thorough job of looking at needs in each community. They can spread out. They make many trips to South Africa and Southern Africa, and they've been all through East Africa and some of West Africa. They're the ones who really do the formal assessment if we're going to go forward with a product. It's a critical step so we can understand what the market might be for whatever is developed.
Right. Sally, this one is for you. Do individuals who benefit from the new diagnostic tests in the communities have access to treatments once you diagnose the condition they have?
Yeah, it's interestingly enough, a lot of times diagnostics actually lag behind treatments, and HIV is a wonderful example in that, the PEPFAR program allowed drugs, medication to be available, but HIV viral load lagged behind. And also initially even just diagnosis of HIV by testing for antibodies. And so as those tests are developed, then people are actually at that time eligible for treatment. And it's the same for tuberculosis. Treating tuberculosis is really pretty inexpensive. It's just antibiotics, but detecting tuberculosis is much more difficult and costly. So it's a real barrier to treatment. And so that's what really inspires me to work on diagnostics.
So just to emphasize everybody that the treatments are there, but you can't treat what you haven't diagnosed, and you need to bring these point of care diagnoses to the community. Matt, can you review again for the audience the countries where we're working and what plans you might have to expand to new countries or even new areas in the current countries we're in?
Yeah, so we've worked in several Southern African countries, but we're really based in South Africa. And in Southern Africa, South Africa is where policy is made, where technologies are developed. So South Africa has been our main focus, or with including the surrounding countries. Since 2012, we've been working in West Africa. Of course, Rob Murphy and his group have been there much longer. But those are the Nigeria, neighboring countries, particularly Mali, are places where we'll be doing some work. I don't know that we have plans to expand. Africa is an enormous place. We do hope that the technology will diffuse into other countries, and I expect that the DASH system will be all over the continent when it's fully deployed.
Yeah, that's great. And just to remind the audience, again, the engineering center of the Institute for Global Health is one of our 10 centers. And just so the audience realizes that the entire institute is involved all around the world. And we do have hopes, especially if we can get funding for the engineering center, to expand ourselves, because there's only one Matt and one Sally, but we can certainly afford to get more faculty and more investigators and more hands on deck if we can expand our funding. So the next one will be for you, Sally. What sort of ethical considerations should we all keep in mind when working in and designing technologies for low and middle income countries?
Great question. So it's really important to understand the setting in which your technology would be located, and to understand what other things the people that would be operating your analyzer might be doing. So we don't want to overburden people with what they call task shifting, where if they used to send the sample out and now we're asking them to test it during the clinical encounter, well, they were already doing something, right? So how can we integrate that into their workflow? And of course, there will also be cultural considerations. It may be for certain types of tests, self-testing might be more accepted than, for example, for sexually transmitted diseases, than for a physician to take the sample. And we have some different projects based on self-testing, which I think it's been demonstrated to be as useful or as effective as testing as a sample collection by a clinician. And it could just allow more people to access healthcare if they're not concerned about this cultural kind of consideration.
Right. Sally and Matt, I think you each can have your own insight on here. Just to remind the audience, what are the type of technologies that the Center for Innovation and Global Health Technology, your center, site, what technologies do you typically focus on? Maybe Sally, you can start.
So, I run the center's product development lab, and we focus primarily on diagnostics, on molecular diagnostics, but also on some lateral flow tests, which is like the kind of home tests that you see for COVID, and also technologies that enable diagnostics. For example, so like the sputum cup or blood collection and plasma separation, things that make a diagnostic work from the patient to the result.
Yeah.
Yeah, I'm going to turn the question around a little bit because it's we don't look for, in general, we don't look for the use of technology. We look for problems, and we will solve the problem with technology if it's appropriate. There are other problems that are not necessarily solved with technology. So as engineers, we're looking to solve the problem, not necessarily to use technology. We've been approached by our collaborators there to develop, for example, an appointment system for patients. When we first got there, patients would line up starting at 4 or 5 a.m. because the day clinics would close by 3, so they had to get in line or they wouldn't get seen. This seemed to our client, our collaborator, to be an inefficient system, and many of the people there thought that these poor and mostly uneducated people wouldn't be able to follow an appointment system. But they were clear. It was obvious immediately that those people were wrong. People don't like waiting at 4 in the morning. We implemented an appointment system. There was a bit of technology involved, I suppose, but it really wasn't a technological solution. Not every solution needs to be technology, and we can fall into that trap. We want a cool device. We want something that's got a microchip in it. We fall into that as engineers because we like the gadgets, but it's not always a device. So we look for projects where we see tractable solutions. Often it involves technology of some sort, but we're not inventing new things. We're designing solutions. That's a different thing. We try to find the best design.
And Matt, can you give an example of maybe a few things that some of your undergraduate students have brought to market, or maybe even if they weren't brought to market, that have been used in some of the communities?
Yeah, so our undergraduates are tremendously talented, enthusiastic, and hardworking, but we're there for about 10 weeks a year. That's how long they have to work on their projects. Nothing is done in 10 weeks, so it's a slow process. The things that have come to market have teams work on them here in our design classes, in South Africa in our program. And one of the difficulties of managing these programs is you have continuous churn of students. Everyone gets to start on a new phase of the project, but they rethink the old phase. So we have a management challenge there, but we have managed to bring a number of products to market. I shouldn't say that. We don't bring products to market. Our collaborators in South Africa bring products to market. So I mentioned the phototherapy Billy blanket, the phototherapy blanket to treat neonatal jaundice. That was brought to market by a company called Infant Trust, who we've collaborated with. Similarly, the sputum cup that I described can be brought to market. In many cases, though, it's a piece of technology. We've had students work on aspects of the DASH system. Sometimes they're looking at market factors. They're not always doing technology. So it's a little bit difficult to say how many things are brought to market, what percentages. They all work on projects that are ongoing. It's a team. So the students are part of the team, Sally's part of the team, I'm part of the team, so are the folks in the other centers. So that's how we keep the projects going. And eventually, a period of 5 or 6 years is what it takes to get anything to market, even something seemingly simple as a sputum cup.
Right. Sally, I know we're going to run short on time, and you could spend an hour talking about DASH. The machine's pretty cool. That video was nice. For those who are younger than us, you might wonder when the YouTube video finished who those other people were. And I think I saw Patty Duke on a couple of them. So I don't think many people know who Patty Duke is. I hardly remember. So that was not part of this presentation, just so we have a little disclaimer there. But, the DASH machine, the reason we have talked about it in even several of our videos is because polymerase chain reaction technology is not easy. It is so sophisticated, we take it for granted. Since COVID, we just take for granted that you can put a swab in someone's nose and pick up one strand of RNA. I mean, imagine how difficult that is. That is a virtual needle in a mountain of hay. And yet you have to find a machine that can do it. And the machines that typically do it take 4 to 8 hours to run and would take up the space of an entire room. So to fit that into a cartridge with the video showing how cool it is inside the machine and the cartridge, that was no small task. So Sally, in a couple of minutes, can you just let us know why you even thought about DASH and how you got that project going?
Well, I think, earlier I mentioned that our goal was to have a test that could be performed at a doctor's office or where a patient's being treated in 15 minutes or less, because there has been studies performed that say that that's really the length of time that somebody can reliably wait for a result. You can imagine that when you've been at the doctor's office and that they're doing a test on you and you're waiting out in the lobby, and you might have another child with you, or you have to go pick up someone at school. It's really difficult to have a patient wait around for too long. But if you can, and then you can treat that patient in the same appointment, there's just so many advantages. One is that you can break transmission. So if the person has, for example, tuberculosis, and you can start them on treatment, then they're not going to go home and necessarily give tuberculosis to their family. So that was really a big motivation for us, was to try to do the test in 15 minutes. And in 2015, Carl Whitworth from the University of Utah, he demonstrated that the speed of PCR was limited by how fast you could get heat into the tube rather than the speed of the enzyme that does the reaction. And so that was kind of a breakthrough that allowed us to start thinking about engineering solutions to how do we get the heat in and out of that tube so that we can cycle, because the cycle part means that you change temperatures, 2 different temperatures, and so you have to get the heat in and have to get the heat out really fast. And so that is what really allowed us to be able to develop DASH to get that fast result. And we were really motivated about being able to provide a result for especially HIV viral load, because when a person comes to collect their medicine, if they're tested and they have detectable viral load, that means their medication isn't working. And maybe they need counseling on how to be sure to take it more regularly, or maybe they need to be tested to see if they need to have their medication changed because they're resistant to that. The virus is resistant to that medication. So, sometimes you need point of care because of the fact that you want the result right in that appointment. And sometimes it's because if you send a sample in to a central lab and get the result back, by the time you get the result back, it doesn't matter anymore. And we saw that in the early days of COVID. I think we can all relate to that one.
Thanks. Perfect. Thanks so much. And Matt, can you just let us know a few things that your next steps for site, what you want to do and how you want to see this grow, your center?
Yeah, so we're interested in doing more work, but we need to attract more people. So that's one of you mentioned some resources. I think a direction we need to go is bringing in some of the excellent water resources people, environmental engineers, to look at some sort of a parallel program in how human health relates to climate change and water. That would and I know there are a number of people at Northwestern interested in joining us in this in a program like that. So we'd like to expand the scope to address some other areas that affect human health. I don't see growing our student programs more than we have. That's a management issue. I would like to try to turn it into a year-round effort. I would love to have a year-round presence in South Africa by Northwestern researchers. So those are the directions we're going to be pursuing in the next few years.
Well, great. Well, thanks, Matt and Sally. We've got 2 minutes left. So just in closing, I want to thank the audience for joining us. I know it's hard to spend an hour listening, but really interesting stuff. And we're just scratching the surface on what engineering can do for global health in low and middle income countries around the world. And I hope you get a little sense from this webinar, which has been different from kind of the COVID-centered webinars we've had over the last few years, that the Institute is really broadly prepared to attack problems from a real multidisciplinary way with a lot of collaboration, because we meet once a week, all of the centers, which is no easy task to get time to do that. We have multiple retreats throughout the year, a lot of cross-pollination even during the week on a different project, with everyone talking to everyone else. And just, I think a lot of the audience has been incredible supporters of our work. People know that I've been involved with this for 16 years. I've done it voluntarily. I've done it because, to put it simply, I think the world has so many challenges, and if we have the ability to make even some impact, we should do it, and we should participate in trying to help the planet, if you will. And I'm not trying to be corny saying that. The world, if you want to get really down to it, has a lot of risks for the future of the world. And global health just has to be something that needs continual work. And look at all the innovation it's going to lead to. Look at the DASH machine, which is a revolutionary technology that was all begun with a small catalyzer grant. For those who forget, our catalyzer program is a $25,000 seed grant to allow people to start work on any project that we think will bear fruit down the road. We right now have about 100 of these catalyzer programs going on in the Institute, 100 different research projects that we funded because of your support. And that's pretty cool to put all that together and imagine spreading that knowledge and those innovations around the world. So thank you for all your support. Thanks for listening. Thanks, Matt. Thanks, Sally. I hope everyone had some fun and learned some things, and we hope to see you again soon. So take care.
Thank you.